Hi, class, and welcome to this week's lecture. Thus far, we have described the main characteristics of neurons, including how their processes come in close contact to form synapses. In this lecture, we will continue to consider the role of the neuron, and in particular, the conduction of communication within a neuron, and how this signal is transmitted to the next neuron. We will then move on to a discussion of how research is conducted in biological psychology. Let's get started. So, there are two stages of this electrochemical action in neurons. The first stage is the electrical conduction of dendritic input to the initiation of an action potential within a neuron. The second stage is a chemical transmission across the synaptic gap between the presynaptic neuron and the postsynaptic neuron of the synapse. To understand these processes, we first need to consider what occurs within a neuron at its steady state, called resting membrane potential. The intracellular fluid inside the cell and the extracellular fluid outside the cell of neurons is composed of a combination of ions, and ions are electrically charged molecules. Cations are positively charged ions, and anions are negatively charged ions. The composition of intracellular and extracellular fluid is similar to salt water, containing sodium, potassium, chloride, and anions. The cell membrane, which is composed of a lipid kind of bilayer of fat molecules, separates the cell from the surrounding extracellular fluid. Proteins span the membrane, forming ion channels that allow particular ions to pass between intracellular and extracellular fluid. These ions are in different concentrations inside the cell relative to outside the cell. And the ions have different electrical charges. Due to this difference in concentration of charge, two forces act to maintain a steady state when the cell is at rest. Diffusion and electrostatic pressure is what this is referred to as. And diffusion is the force on molecules to move from areas of high concentration to areas of low concentration. And electrostatic pressure is the repulsive force between similarly charged ions and the attractive force between oppositely charged ions. Remember the saying, opposites attract. So there is a membrane potential at which the force of diffusion is equal and opposite to the force of electrostatic pressure. This voltage is called the equilibrium potential, and it's the voltage at which no ions flow. Since several ions can permeate the cell's membrane, the baseline electrical charge inside the cell compared with outside the cell is referred to as the resting membrane potential. And it's based on the collective drive of force on several ions. Relative to the extracellular fluid, the membrane potential of a neuron at rest is negatively charged at approximately negative 70 millivolts. These voltages are tiny compared to those of batteries and electrical outlets, which are measured in volts, not millivolts, and range from 1.5 to 240 volts. So let's see how these two forces, diffusion and electrostatic pressure, act on the four groups of ions that I just mentioned. Anions are highly concentrated inside a cell and contribute to the negative charge of the resting membrane potential. Diffusion and electrostatic pressure are not forces that determine a concentration because anions are impermeable to the cell membrane. There are no ion channels that allow for anions to move between the intracellular and extracellular fluid. Potassium is another one to consider. And the cell membrane is permeable to potassium at rest, but potassium remains in high concentrations inside the cell. Diffusion pushes potassium outside the cell because it is in high concentrations inside the cell. However, electrostatic pressure pushes potassium inside the cell because of the positive charge of potassium. And that positive charge is attracted to the negative charge inside the cell. In combination, these forces oppose one another with respect to potassium. With chloride, the cell membrane is also very permeable to it, and chloride, especially chloride at rest. But chloride remains in high concentrations outside the cell. Diffusion pushes chloride inside the cell because it is in high concentration outside the cell. However, electrostatic pressure pushes chloride outside the cell because of the negative charge of chloride, and that's attracted to the positive charge outside the cell. So these forces on chloride oppose one another. Sodium is another one, and the cell membrane is not very permeable to sodium at rest. Diffusion pushes sodium inside the cell because it is in high concentrations outside the cell, but electrostatic pressure also pushes sodium inside the cell because of the positive charge of sodium and how that's attracted to the negative charge inside the cell. So both of these forces push sodium inside the cell. However, sodium cannot permeate the cell membrane and remains in high concentrations outside the cell. The small amounts of sodium inside the cell are removed by a sodium-potassium pump, which uses the neuron's energy and the adenosine triphosphate ATP to pump sodium neurons out of the cell in exchange for bringing in potassium inside of the cell. So now that we've considered what occurs in a neuron at rest, I think it's important for us to consider what changes occur to the resting membrane potential when a neuron receives input from the presynaptic terminal button of another neuron. So our understanding of electrical signals or potentials within a neuron results from the seminal work of Hodgkin and Huxley that began in the 1930s at a well-known marine biology lab in Woods Hole, Massachusetts. Their work, for which they won the Nobel Prize in Medicine in 1963, resulted in the general model of electrochemical transduction. Now, Hodgkin and Huxley studied a very large axon in a squid, a common species for that region in the US. The squid's axon is roughly 100 times larger than axons in the mammalian brain, like ours, making it much easier to work with. Activation of the giant axon, whose large size allows for rapid electrical signal transmission, triggers a swift withdrawal response that enables the squid to escape from predators. Now, while studying this species, Hodgkin and Huxley noticed that if they applied an electrical stimulus to the axon, a large transient electrical current conducted down the axon. This transient electrical current is known as an action potential. Now, an action potential is an all or nothing response that occurs when there is a change in the charge or potential of the cell from its resting membrane potential in a more positive direction, which is a depolarization. An all or nothing response parallels the binary code used in computers, where only two possibilities exist, 0 or 1. There are no intermediate values. 0.5, for example, doesn't exist in binary code. Similarly, an action potential either occurs or it doesn't, with no partial activation. This all or nothing principle applies to both binary code and action potentials. They are either triggered fully or not at all. And there's a specific membrane potential that the neuron must reach to initiate an action potential. This membrane potential, called the threshold of excitation, is typically around negative 50 millivolts. If the threshold of excitation is reached, then an action potential is triggered. So, how is an action potential initiated? At any one time, each neuron may receive hundreds of inputs from the cell that synapses with it. These inputs can cause several types of fluctuations in the neuron's membrane potentials. The first is excitatory postsynaptic potentials, or EPSPs, a depolarizing current that causes the membrane potential to become more positive and closer to the threshold of excitation. Another is inhibitory postsynaptic potentials, or IPSPs, a hyperpolarizing current that causes the membrane potential to become more negative and further away from the threshold of excitation. These postsynaptic potentials, EPSPs and IPSPs, summate or add together in time and space. The inhibitory IPSPs make the membrane potential more negative, but how much so depends on their strength. The excitatory EPSPs make the membrane potential more positive. Again, how much more positive depends on their strength. If you have two small EPSPs at the same time, at the same synapse, the result will be a large EPSP. If you have a small EPSP and a small IPSP at the same time and same synapse, then they'll cancel each other out potentially. Unlike the action potential, which is an all or nothing response, IPSPs and EPSPs are smaller and graded potentials varying in strength. The change in voltage during an action potential is approximately 100 MV, or millivolts. In comparison, EPSPs and IPSPs are changes in voltage between 0.1 to 40 millivolts. And they can be different strengths or gradients and are measured by how far the membrane potentials diverge from the resting membrane potential. The concept of summation can be confusing, right? And I think that it's important to go over. So, as a child, for example, I used to play a game with a very large parachute where you would try to knock balls out of the center of the parachute. This game illustrates the properties of summation. In this game, a group of children next to one another would work together to produce waves in the parachute in order to cause a wave large enough to knock the balls out of the parachute. The children would initiate the waves at the same time and in the same direction. The additive result was a larger wave in the parachute, and the balls would bounce off and out of the parachute. However, if the waves they initiated occurred in the opposite direction or with the wrong timing, the waves would cancel each other out and the balls would remain at the center of the parachute. EPSPs or IPSPs in a neuron work in the same way. They either add or cancel each other out. So if you have two EPSPs, then they sum together and become a larger depolarization. Similarly, if two IPSPs come into the cell at the same time, they'll sum and become a larger hyperpolarization of the membrane potential. However, if two inputs were opposing by one another, moving the potential in the opposite direction, such as EPSPs and IPSPs, their sum would cancel each other out. At any moment, each cell is receiving mixed messages, both EPSPs and IPSPs. If the summation of EPSPs is strong enough to depolarize the membrane potential to reach the threshold of excitation, then it initiates an action potential. The action potential then travels down the axon away from the soma until it reaches the end of the axon, the terminal button. In the terminal button, the action potential triggers the release of neurotransmitters from the presynaptic terminal button into the synaptic gap. These neurotransmitters, in turn, cause EPSPs and IPSPs in the postsynaptic dendritic spines of the cell, or of the next cell. The neurotransmitter released from the presynaptic terminal button binds with ionotropic receptors in a lock and key fashion on the postsynaptic dendritic spine. Ionotropic receptors are receptors on the ion channels that open, allowing some ions to enter or exit the cell depending upon the presence of a particular neurotransmitter. The type of neurotransmitter and the permeability of the ion channel it activates will determine if an EPSP or IPSP occurs in the dendrite of the postsynaptic cell. These EPSPs and IPSPs summate as described previously, and the entire process occurs again in another cell. So I spoke about how previously which ions are involved in maintaining the resting membrane potential. And not surprisingly, some of the same ions are involved in the action potential. When the cell becomes depolarized or more positively charged and reaches the threshold of excitation, this opens a voltage dependent Na plus channel. Okay. A voltage dependent channel is a channel that opens, allowing some ions to enter the cell, or to enter or exit the cell, I should say, depending upon whether the cell reaches a particular membrane potential. When the cell is at rest, or resting membrane potential, these voltage dependent Na plus channels are closed. As we learned earlier, both diffusion and electrostatic pressure are pushing Na positive inside the cells. However, Na positive cannot permeate the membrane when the cell is at rest. Now that these channels are open, Na plus rushes inside the cell, causing the cell to become very positively charged relative to the outside of the cell. Now, this is responsible for the rising or depolarizing phase of action potential. Now, just a reminder, when we're talking about Na+, we're talking about sodium. And we're going to be talking about K+, which is potassium, right? So just a little reminder there. The inside of the cell becomes very positively charged, so about positive 40 millivolts. At this point, the Na plus channel, the sodium channels, close and become refractory, which means that the Na plus channels, the sodium channels, cannot reopen again until the cell returns to the resting membrane potential. Thus, a new action potential cannot occur during the refractory period. The refractory period also ensures the action potential can only move in one direction down the axon, away from the soma. As the cell becomes more depolarized, a second type of voltage-dependent channel opens. And this channel is permeable to K+, or potassium. With the cell depolarized very positively relative to the outside of the cell, the high concentration of potassium, or K+, within the cell forces of both diffusion and electrostatic pressure drive K+ outside of the cell. The movement of K+ out of the cell causes the cell potential to return to the resting membrane potential, the falling or hyperpolarizing phase of the action potential. A short hyperpolarization occurs partially due to the gradual closing of the K+ channel. With the Na+ closed, the electrostatic pressure continues to push K+ out of the cell. In addition, the sodium-potassium pump is pushing Na+ out of the cell. The cell returns to the resting membrane potential, and the excess extracellular K+ diffuses away. This exchange of Na+ and K+ ions happens very rapidly, in less than 1 millisecond. And the action potential occurs in a wave-like motion down the axon until it reaches the terminal button. And only the ion channels in close proximity to the action potentials are affected. So earlier I spoke about how axons are often covered in myelin. That was in previous lectures. So it's important for us to consider how myelin, the myelin sheath, speeds up the process of action potential. So there are gaps in the myelin sheath called nodes of Ranvier. And the myelin insulates the axon and does not allow any fluid between the myelin and the cell membrane. Under the myelin, when sodium potassium channels open, Na plus and K plus channels open, no ions flow between the intracellular and extracellular fluid. This saves the cell from having to expend the energy necessary to rectify or regain the resting membrane potential. Remember, the pumps need ATP to run. So under the myelin, the action potential degrades some, but still has a large enough potential to trigger a new action potential at the next node of Ranvier. Thus, the action potential jumps from node to node. This process is known as saltatory conduction. Saltus means jump in Latin, so that's where the name comes from. In the presynaptic terminal button, the action potential triggers the release of neurotransmitters, and neurotransmitters cross the synaptic gap and open subtypes of receptors in a lock and key fashion. So depending on the type of neurotransmitter, an EPSP or IPSP occurs in the dendrite of the postsynaptic cell. Neurotransmitters that open Na plus or calcium Ca plus channels cause an EPSP. An example is in the NMDA receptors, which are activated by glutamate, the main excitatory neurotransmitter in the brain. In contrast, neurotransmitters that open Cl negative or K plus channels cause an IPSP. And an example is the gamma aminoburic acid or GABA receptors, which are activated, GABA, as they're often referred to, which are activated by GABA, the main inhibitory neurotransmitter in the brain. Once EPSPs and IPSPs occur in the postsynaptic site, the process of communication within and between neuron cycles, between neurons cycle on, I should say. A neurotransmitter that does not bind to receptors is broken down and inactivated by enzymes or glial cells, or is taken back into the presynaptic terminal button in a process called reuptake, which I'm going to discuss further in our lecture on psychopharmacology, because modulating reuptake is a big part of how modern antidepressants work. Keep in mind, you've probably heard in your other psych classes about SSRIs, selective serotonin reuptake inhibitors, right? So it modulates reuptake. It's very interesting. We're going to get into that in later lectures. Now, it's important to understand that biological psychology is a broad and diverse field that consists of many different approaches, including neuroscience, neuropsychology, cognitive neuroscience, behavioral genetics, and psychopharmacology, just to name a few, but also dovetails heavily with psychiatry and clinical psychology. So with so many different approaches touching it, it's not surprising that the research methods used in biological psychology are also broad and diverse. So we're going to be taking the rest of this lecture to start talking about biological psychology research methods. So what do these look like? Well, techniques range from low-level approaches like recording the activity of a single neuron and dissecting animal brains to high-level cognitive testing and brain imaging in humans. We've learned about brain function from case studies of brain damage and from technical marvels such as high-resolution brain imaging and optogenetics, in which cells in animal brains are genetically manipulated so that they can be controlled by specific wavelengths of light while the animal is awake. Each different technique in biological psychology has strengths and limitations and can be used to study distinct types of questions. When establishing the specific function of a particular brain area, the strongest evidence comes from converging evidence, whereby multiple studies using different methods report similar or converging findings, right? If you've taken my research methods class, you've seen how important mixed methods designs are. This is an example of that. In this portion of our lecture, we're going to cover some of the major research methods in biopsychology, psychobio, biological psychology, however you want to refer to it, and how the methods converge to help us understand how the 3-pound human brain gives rise to our thoughts, actions, perceptions, feelings, and emotions. Now, early insights into brain and behavior links emerged from cases of brain damage. And prominent examples from the 19th century include Phineas Gage, who you probably heard about in your intro psych course, and the patients of physicians like Paul Braca and Karl Wernicke, who you certainly heard about if you took my intro psych course. In the mid-1800s, the railroad worker Phineas Gage was responsible for setting explosive charges to blast through rock for railroad tracks. Using a 1 meter long tamping iron, he would tamp down on the charges built on, kind of built into the ground. But on one September afternoon, a spark set off the explosive prematurely. The tamping iron shot through the air like a rocket, entering the side of Gage's face, passing behind his left eye, and exiting the top of his head, landing 80 feet away. Gives you an idea of the intense force of this blast. Amazingly, he survived. And the accident was something that became psychological lore for, one might argue, the rest of time. He was even able to talk and walk away from it. And that's a big reason why. Though he lost a portion of his left frontal lobe, he lived for another 12 years without apparent impairment in speech, motor abilities, memory, or intelligence. Super wild. However, what's fascinating about this incident from a psychobio perspective is that Gage's personality changed drastically as a result of the accident. Before the accident, Gage had been responsible, socially well adjusted. But afterwards, he became irreverent, vulgar, impulsive. He didn't follow social conventions, and he had difficulty executing plans. So in another example from the 1800s, the French physician Paul Braca found that some patients who were unable to produce speech had brain damage to their left inferior frontal cortex. One of Braca's patients, nicknamed Tan, could only produce the single syllable tan repeatedly. After Tan's death, Braca discovered a major lesion on Tan's left frontal lobe. Another patient of Braca's was also severely aphasic, meaning that he couldn't produce fluent speech and had brain damage to the same portion of his left frontal lobe. This led Braca to surmise that speech production was localized to this region of the left inferior frontal lobe. Around the same time, German physician Karl Wernicke discovered that damage to a different brain region, the left superior temporal lobe, was associated with impaired speech comprehension. So together, this double dissociation showed that brain damage to specific locations are linked to specific types of behavioral impairments, with one area being referred to as Braca's area and the other referred to as Wernicke's area. So these and countless other examples show that brain damage can lead to behavioral and cognitive impairments. Such cases demonstrate localization of function, that certain brain regions perform specific functions, like the previous examples of impulse control, speech production, and perception. However, studies with human brain damage are a limited tool for understanding the brain. For example, when a patient suffers brain damage from force, trauma, a tumor, or stroke, or a lesion, I should say, like Braca's patient Tan, that brain damage is unique to that patient, making it difficult to extrapolate or generalize to others' brains unless you see the same kind of lesion or damage causing the same kind of impairments again and again. And it's rarely just and damage is rarely just confined to one brain area, unless it's intentionally done in an experimental form, which is unethical, right? Which causes us to rely on organic accidents, making it difficult to isolate the roles of specific brain structures. And like I mentioned before, alluded to before, creating controlled and localized damage to human brains in laboratory experiments just isn't possible because of ethical constraints. So researchers have resorted to creating carefully controlled brain damage or lesions in laboratory animals, such as mice and rats. Not the most pleasant thing in the world, especially for those of us who care a lot about animals' well-beings. Now, invasive techniques such as lesioning brain regions or implanting or recording electrodes in the brain, like you see on this mouse in the graphic right here, are commonly used in psychobio research with non-human animals. For the experimental procedure, the animal undergoes surgery using a stereotaxic unit right here, that you can see on this graphic on the slide, that allows precise positioning in the brain. The stereotaxic unit has two main parts: a head holder to immobilize the head, and an electrode holder, which holds the device to be inserted. After the animal is anesthetized, its head is immobilized in the head holder. To locate target positions in the brain, researchers use a stereotaxic atlas, kind of like a road atlas, but showing locations of the brain regions instead of roads and cities. The researcher then drills a small hole and performs the surgery. In some studies, researchers will lesion or remove a part of the brain. This is called an ablation. And the surgery may use a surgical knife, a current passing electrode, or an aspirating suction pipette. After the target region is carefully removed or inactivated, the animal's behavior is tested to determine if the function of the lesion structure has been affected. For example, researchers may remove subcomponents of the amygdala or hypothalamus and test how behavior changes. Studies will often include a control condition in which the control animals receive a sham lesion, wherein they undergo a similar surgical procedure but don't receive a lesion. This control condition allows researchers to more confidently interpret the change in behavior and determine whether it stems from the lesion brain area rather than the handling, anesthesia, or other ancillary procedures. In other words, the direct manipulation of the brain coupled with the control condition allows researchers to make some causal claims, something that cannot be done with correlational research methods. After the animal's death, its brain is often cut into thin slices and stained to highlight neuronal structures. These slices are then examined microscopically to verify the extent and precise location of the lesions. As an alternative to permanent lesions, researchers can lesion brain regions temporarily. This can be achieved by cooling the target brain tissue or injecting it with an anesthetic. Additionally, electrical stimulation from implanted electrodes can temporarily inactivate or activate targeted brain regions. In addition to manipulating brain function by damaging or stimulating brain regions, researchers learn about brain function by implanting recording devices directly in an animal and in their brains to listen in on brain activity. The animal undergoes a stereotaxic surgery wherein a recording device or electrode is inserted into the target area, then fixed into a position on the skull. So different types of invasive electrophysiological recordings include intracellular recording, or intracellular unit recording, I should say, wherein a microelectrode is inserted into a single neuron to measure its electrical activity. extracellular recordings that pick up the firing of one nearby neuron, a single unit recording, or several adjacent neurons, a multi-unit recording, and also invasive electroencephalography, or EEG, wherein a large electrode picks up the electrical brain activity of a swath of nearby neurons. These electrodes detect neural activity as the animal performs a task, providing insight into the relationship between neural activity and behavior. Directly recording from within human brains for research is rare, as invasively opening the skull cannot be ethically justified for research purposes alone. However, that being said, some patients who are already undergoing surgery for medical procedures, such as treatment for Parkinson's disease or epilepsy, might have electrodes placed directly in their brains if they consent to it. They occasionally participate in research. Such intracranial, meaning within the skull, electroencephalography, or IEEG, or electrocorticography, ECOG, is a valuable research tool because these direct recordings offer exceptionally clear and precise signals from the human brain. However, these patients are rare, and the placement of electrodes is determined by the neurosurgeon and the medical needs of the patient rather than the research question. So, advances in technology have led to increasingly sophisticated brain recording techniques. Just like X-rays allow us to peer inside the body, neuroimaging techniques enable us to view the working brain. Biopsychology, or psychobio, employs various non-invasive methods to study the human brain functioning, each with its own advantages and disadvantages. It's crucial to recognize that each technique offers a unique perspective on brain activity. Understanding these methods is essential, as our comprehension of the brain is closely tied to available technologies. As new technologies develop, we can anticipate an even deeper understanding of the brain. Electroencephalography, or EEG, measures the electrical activity of the brain and has been used for a century. When large populations of neurons are active, they create small electrical voltages that pass through the skull and scalp. Electrodes on the participant's head pick up the voltages, which are amplified and recorded. Researchers can record the voltages from brain activity as the participant performs a task. The electrical signals generated in the brain become distorted as they travel through brain tissue, skin, and bone. As a result, researchers can only approximate the original source of these signals within the brain, and this uncertainty is especially pronounced for signals originating deep within the brain, even with advanced tools such as structural brain scans. High-density electrode coverage from a cap with 256 electrodes, like the one that you see on the slide right here, and sophisticated analysis algorithms for localization. Thus, EEG's spatial resolution, its ability to pinpoint where activity occurs in the brain, is relatively low. Conversely, EEG's temporal resolution is excellent and indicates when something happens in the brain within millisecond precision. EEG's superior temporal resolution makes it ideal for examining the brain's rapid response to a stimulus event, or the event-related potentials, ERPs. In a typical event-related potential experiment, ERP experiment, researchers might play a visual or auditory event like a word and measure the corresponding voltage changes that unfolds in the brain over the next few hundred milliseconds. The amplitude, timing, and topography, or position of the EEG signal, captures the underlying neural and mental processes. The high temporal resolution and continuous recording of EEG allows it to capture different frequency brain waves, such as theta waves oscillating at 4 to 7 hertz, or cycles per second, or alpha waves at 8 to 13 hertz, beta waves at 14 to 30 hertz, etc. Brain oscillations reflecting the combined activity of millions of neurons capture attentional and conscious states. These oscillations play a crucial role in information encoding and attention modulation in the brain. In addition to research applications, EEGs play a crucial role in signal diagnosis, particularly in identifying epilepsy, seizures, and sleep disorders. Um, Magnetoencephalography, or MEG, is similar to EEG, but instead of electrical signals, MEGs pick up the weak magnetic fields generated by the flow of electrical charge associated with neural activity. Because the magnetic fields generated by brain activity are so small, special rooms are needed to shield out magnetic fields in the environment so that MEG sensors pick up magnetic fields from neural activity without environmental contamination. Similar to EEG, MEG also has excellent, within a millisecond, temporal resolution. The spatial resolution of MEG is far better than EEG because magnetic fields pass relatively unchanged through hard and soft tissue, so are not distorted by skull and scalp tissues and bone. Despite MEG's excellent spatial and temporal resolution, though, it's less widely used than EEG because, as is often the case in limiting research, it's much more expensive and complex to operate. Now, I've mentioned MRIs in passing before in the first couple of lectures, and I mentioned structural MRIs and fMRIs. We're going to go into a little bit more detail when it comes to tools of cognitive neuroscience, so brain imaging techniques. And the most commonly used brain imaging modality today is an MRI, magnetic resonance imaging. You've all very likely heard of it. An MRI machine can effectively produce different types of scans. High-resolution images of brain structures, structural MRIs or SMRIs, and brain function, functional MRIs or fMRIs. MRI scanners may be expensive, noisy, and claustrophobic to some, but they're harmless and painless and are powerful and prevalent tools for illuminating brain structures and functions. Magnetic resonance scanners use a strong magnetic field that is 60,000 times stronger than the Earth's magnetic field. As a person lies very still in the scanner, the magnetic field forces protons in their body to align. Pulsations of low energy radio frequencies cause the protons to change their spin. As the radio frequency is turned off, these protons return to their aligned state and give off energy that is detected by MRI sensors. The timing and amount of energy released as protons realign with the magnetic field vary based on the tissue type. But this variation allows clear differentiation between the brain's white matter, gray matter, cerebrospinal fluid, bone, blood, and other tissues. Now, structural magnetic resonance imaging, which we talked about a few lectures ago in passing, creates detailed images of brain structures within millimeter resolution. And the high resolution 3D images might show the brain's gray matter and white matter in voxels, like 3D pixels that are 1 millimeter by 1 millimeter by 1 millimeter cubes. And researchers may use these images to compare the size of brain structures across different groups. For example, are areas associated with pleasure smaller in individuals with depression, or are areas controlling finger movements larger in string musicians compared to vocalists or trombonists? These structural images can also enhance the spatial accuracy of functional magnetic resonance imaging, so fMRI measurements. There's also diffusion tensor imaging, or DTIs, and that's a variant of structural MRIs that focus on myelinated axon pathways in the brain. DTI imaging is highly sensitive to the movement of water molecules in the brain. And because water moves differently along myelinated axons in the brain, DTI can map out large white matter tracts that are like superhighways that connect distant brain regions, like the corpus callosum, a white matter fiber tract that connects the left and right hemispheres, or the oculate facilius, a bundle of axons that connects the Broca's area and Wernicke's area, also known as the language loop. Now, DTI can be used to examine white matter integrity in diseases such as MS or multiple sclerosis, or to observe brain plasticity, the ability to adapt and learn new skills when learning a new talent or new skill like juggling, for example. Now, fMRIs, functional magnetic resonance imaging, uses the same MR scanners, magnetic resonance scanners. But instead of capturing high resolution snapshots of brain structures, it measures brain function or activation while a subject performs some task. As a brain region becomes more active, it uses oxygen and causes an inflow of oxygenated blood to that region over the following few seconds. fMRI measures the change in the concentration of oxygenated hemoglobin, which is known as the blood oxygen level dependent or BOLD signal, BOLD. From the BOLD signal, researchers infer neuronal activation in that brain region. It's important to note that if the fMRI, does not directly measure neuronal activity. Because cerebral blood flow is coupled with neural activation, researchers can map activation while people in the scanner perform tasks like reading, speaking, viewing images of faces, places, recalling memories, etc. In this way, fMRI provides evidence of localization of function and which areas are active during specific tasks. fMRIs have high spatial resolution, and the activation maps in a typical fMRI study in such a way that it consists of cubic voxels that are a few millimeters on each side. However, the temporal resolution of MRIs are quite poor, and it typically takes a snapshot of brain activation averaged over a 2 to 3 second window. In addition to measuring the blood oxygenated level dependent signals, the BOLD responses, while subjects perform a single task, fMRIs can measure the subject's brain activation over many minutes while they perform no task, so-called resting state scans, wherein they might lay in the scanner for 10 minutes while instructed to not do anything in particular. Such recordings have shown surprisingly correlated spontaneous fluctuations of brain regions that can be far apart from one another in the brain. Regions with highly correlated activation work together in the same large-scale distributed network. The brain has several large-scale networks, including sensory motor, attention, control, default mode, and limbic networks. And while fMRI is popular and powerful, and people find the images, the pretty images convincing, they're correlational and don't fully explain the causal role of brain regions in determining mental processes. This is an important example of why it's essential to rely upon converging evidence from multiple sources. As an example, correlational fMRI data coupled with causal experimental data from lab animals can be particularly robust. Also, to address some of the limits of correlational research, researchers are developing techniques that can directly modulate brain activity. So finally, neuroimaging studies focus on correlations between brain activity and behavior and can't establish a causal role of a brain region in determining behavior, as I mentioned. So to establish a causal rather than correlational relationship, we need to alter brain functioning to observe resulting changes in behavior. Lesions are one way to modify brain structures and can reveal a causal relationship. Like when losing a brain region leads to a loss of function, that brain area is deemed necessary or involved in that function. However, invasive lesions can only be done in animals, at least from a purely experimental perspective, which differ from humans in key ways. Lesions in the human brain can only be studied in patient populations, that is, after a patient experiences brain damage from a stroke or other injury. New technologies allow researchers to temporarily and non-invasively modify human brain function. Transcranial magnetic stimulation, or TMS, is a form of brain stimulation that uses magnets to alter brain activity. Researchers place a magnetic coil over the scalp and apply magnetic current that stimulates the neurons below the magnetic coil. And in fact, this technique is used for treatment-resistant depression as a treatment in and of itself. But from a research perspective, depending on the type and rate of magnetic impulses, TMS can temporarily turn off or turn on the brain area under the coil. In research domains, researchers might temporarily turn off or turn on parts of the frontal lobe and look at subsequent feelings of craving or emotional processing. And like I said, TMS is also used as an effective clinical treatment. Now, transcranial direct current stimulation is a bit different. That's TDCS, similar to TMS, except in some ways, but it's different in the way that it uses an electrical current directly rather than inducing it with magnetic pulses via small electrodes on the skull. A brain area is stimulated by a low current equivalent to a AA battery for an extended time. When combined with cognitive training, TDCS has been shown to improve many cognitive functions such as mathematical ability, memory, attention, and coordination. We also have gene knockout, which is a genetic technique used in animals wherein researchers remove or inactivate a specific gene. This allows researchers to study the function of that gene in a living organism and its effects on the phenotype, the physical expression of that gene. Gene knockout is considered a loss of function mutation. What function is lost after knocking out a specific gene? Well, gene knockouts are used in many organisms, including fruit flies, zebrafish, and mice. Studies using knockout mice, as they're referred to, have been extremely valuable in understanding the role of genes in brain development and neurological diseases. Now, gene knockout is a type of technique where or gene knock-in, I should say, is a type of technique that is similar. It's related to gene knockout. But instead of removing the gene knock-in inserts a gene. Gene knock-in is considered a gain of function mutation. Well, what function is gained after inserting a gene? Well, we have, it really depends on what they're trying to accomplish. Now, Brainbow is another innovative transgenic technique. Transgenic means transferring genes from one organism to another, that inserts genetic material to label individual neurons with distinct colors and produce detailed neural maps. In Brainbow, green fluorescent protein, a protein found in jellyfish and corals that exhibits bright green fluorescence, is generally genetically modified to produce different colors. These various fluorescent proteins are then inserted into individual neurons in different ratios, flagging each neuron with a unique color. Brainbow has enabled the simultaneous mapping of hundreds of neurons and allows scientists to trace the intricate connections between neurons. Thus, Brainbow has been groundbreaking for the field of neural connectomics, which studies the organization of neural networks. This technique provides striking images of neurons and highlights psychobio research at the molecular and cellular level. And finally, optogenetics is an especially exciting technique for manipulating brain activity in non-human animals. Optogenetics uses light to control specific populations of neurons in living animals. And for the neurons to be sensitive to light, researchers genetically insert light-sensitive proteins taken from algae into a specific type of neuron. After inserting tiny optical fibers into the animal's brain, researchers can turn on the light to excite or inhibit these specific cells. Scientists have used the ability to control the activity of specific neuronal populations to investigate their role in learning, memory, decision-making, addiction, movement, and many other areas of active research. In sum, the various research techniques used in psychobio each have their strengths and weaknesses in terms of spatial resolution, temporal resolution, ease of use, invasiveness, cost, precision, ethics based on population, etc. Using the different tools in a complementary manner provides converging evidence for understanding how the brain works. Well, that'll do it for this week. Be sure to turn to your discussion question and answer the discussion in full, both responding to the question and providing two substantive responses to the answers of your classmates. I look forward to reading them all. And as always, feel free to ask any questions via email at the campus inbox. Take good care and be well. And I'll look forward to speaking with you all next week.